my name is Angie, and you're going to learn about CRISPR today, so let's get going.
So what is CRISPR? CRISPR is an enzyme that can replace or edit genes by cutting the specific genome in the target DNA. For the bacteria, CRISPR is a defense mechanism, so bacteria uses CRISPR to protect itself from a viral attack. Bacteria gets attacked by a virus? How? That's a very good question, because when a virus gets attached to the cell membrane of the bacteria, as we can see right here, it can easily inject its RNA into the cell. So we can see right here the capsid dissolves in the cell membrane, releasing its two enzymes and RNA. So now the viral RNA uses the help of reverse transcriptase enzyme to become single-stranded and becoming double-stranded DNA at the end. When it's done, this integrase enzyme right here grabs the viral DNA into the bacteria DNA and cuts out a fragment of the bacteria DNA to insert the viral DNA. And right here, as we can see it right here, inserted its viral DNA into the bacteria DNA. So now they're together. And according to the lysogenic system, when the viral DNA integrates into the host DNA, the viral DNA will be replicated with the cell passing the viral DNA. The viral DNA, we can see right here, the oranges. It will be replicated with the cell membranes. And we do not want that. That's why we have to use CRISPR in order to save the bacteria. So to make the CRISPR enzyme to work and do its job, we have to add some ingredients. So we have to have tracker RNA, we have to have cRNA, and we have to have Cas9 protein. We have to include cRNA because it carries the previous viral attack's little segment of the virus DNA that can tell the protein where to go and find the foreign DNA. And then the tracker RNA base pairs with the cRNA to form guide RNA. Basically means it will take us where we need to cut. So Cas9 protein uses the tracker RNA portion of the guide as a handle, while the cRNA spacer sequence directs the complex to matching a viral sequence. Not only they have to find the foreign DNA, they also look for PAM sequence right here that tells the enzyme that it is okay to make the cut. Without PAM sequence, the CRISPR wouldn't know which fragment they have to cut, so they might even cut their own CRISPR locus. However, once they make the cut, there will be open space between the bacterial DNA. Thus, we have to flood template DNA that is complementary to the bacterial DNA, and this process is called HDR. Once we take out the mutated gene, we flood cell with desired sequence DNA, and the cell will use the correct copy to fix the cut made from the CRISPR right here. Earlier, I mentioned about CRISPR locus. CRISPR locus is important because it saves the little fragments of the viral DNA that we just cut out from the bacterial DNA, and it saves it. So when the same viral DNA comes back, they're able to recognize it and able to fight back. And that's how we save the bacterial cell from the virus.
CRISPR is very interesting and important enzyme that in the future we are hoping to be able to cure cancer or any type of disease using CRISPR, just cutting the mutated gene and replacing it with the right DNA.